Hello my dear students, and welcome you all to another video lecture on my YouTube channel. As you can see from the title written over here, today we are going to discuss another very important topic, that is genotype and phenotype. To begin with, let me ask if you have ever been surprised by the fact that a zygote as a single cell is capable of forming the whole body of the organism. Can you believe that a zygote as a single cell forms 200 different types of cells and 11 most complex organ systems of the human body? Without having any capacity to think, the zygote has the potential to design most complex organ on this planet, that is the brain of humans. To all such queries and surprising facts, you can find the most satisfactory explanation only in genetics. Why? Because it is all related with the genes. Yes, we are what our genes are. Our structure, our function, our behavior, everything related to us as an individual living organism. We are being controlled by the genes. When we begin our life as a single zygote, the only property we receive from our parents is the genetic material. And it is this genetic material that guides the formation of our all cells, all tissues, all structures, and all our organ systems. Most common terms which are related with this genetic material and subsequent expression of this genetic material used in genetics is this genotype and phenotype. Today in this lecture, we are going to discuss the meaning of the genotype and phenotype, and we'll also try to understand how these two terms are linked with each other. Genotype term in genetics has been used to describe the traits at multiple levels. Either it has been used to describe a single trait, or it has been used to describe multiple traits, or it has been used to describe all the phenotypes of an individual. Most commonly, it has been used with respect to the single trait. And with respect to single trait, it can be defined as the set of alleles in an organism on a single locus related to single trait. That is, what is the set of alleles present in an organism on a single locus is related to a single trait. That is, the set of alleles which are present in an organism located on the single locus of homologous chromosomes, and these alleles are controlling or related to single trait. And what is the characteristic, what is the phenotype, what is the trait? This has already been discussed in the previous lecture. As discussed in the previous lecture, let us again suppose that this is the homologous pair. That is, this chromosome has been received from the father through the male gamete, and this chromosome has been received from the mother through the female gamete. And these two chromosomes are homologous. And by homologous chromosomes, we mean these chromosomes are having same structure, same size, and same set of genes controlling the same set of characteristics. Now, we are going to explain what it means by the single locus related to the single trait. And that set of alleles which are located on the single locus and controlling the single trait is called as the genotype. So let me try to explain it. For example, we have at this location one gene, and on this location we are having the another gene, and this is called as the locus, as I have described it in the previous lecture. Now this is one locus, and here we have another locus. So at this position, this set of genes are controlling a one characteristic, and that characteristic is, for example, the shape of seed. Both these alleles are controlling the shape of the seed. Now these two genes present on the single loci are two alleles. This is one allele and this is another allele. Similarly, at this location we have at this locus two alleles, and these two alleles might be controlling another characteristic. Say for example, it is the color of seed. Okay, Similarly, we have for every characteristic, we have two alleles. One allele through this chromosome is received from the father, and one allele through this chromosome is received from the mother. Now, these two alleles may be controlling the same trait. Trait in this sense is either round or it is wrinkled. And similarly, in this case, the trait may either be yellow or it may be green. So, I have already discussed it that when we are discussing these traits, then we are referring to the alleles. When we are referring to this characteristic, we are referring to the gene. Now we have two alleles located at the same locus, and they are controlling the same characteristic, and they may be displaying two contrasting traits. And these alleles, as you remember, in the Mendelian genetics, we have a specific notation for them. For example, here, if this allele is controlling the round trait, then we will represent it as capital R. And this allele right here on the same locus, if it is controlling the wrinkled trait, then we will denote it by small r. Now, at this locus, we are having two alleles. One is capital R and another is small r. And another case may arise that here it may be small r or here it may be capital R. Or another case may be at both these locations we have small r, or we have capital R at both these locations. That is, there are three possibilities. That is, we are having two alleles at this locus which are being represented by capital R and small r. Now there are three possible combinations. Either it may be capital R and capital R, or it may be the capital R and small r, or it may be the small r and small r. So this is the set of alleles. Now with respect to these set of alleles, we are expecting accordingly the phenotype or trait. So in this case, as you know, we have phenotype that is round. Okay, again in this case we have phenotype that is round, and in this case we have the phenotype that is wrinkled. Now this set of alleles with respect to this phenotype is called as the genotype. This set of alleles present at this location with respect to related to this phenotype is called as the genotype. And similarly, this set of alleles present at this locus related to this phenotype is called as the genotype. Now you can understand that what it means by set of alleles present in an organism on single locus related to single trait. That is related to single trait, not related to the single characteristic. I again repeat it that what is characteristic, what is trait, what is phenotype, I have already explained it in the previous lecture. So here this set of alleles related with this phenotype is the genotype of this individual, and this genotype is related to the single trait. Similarly, in this case we have two alleles present, and these two alleles, this set of alleles located at this location are controlling the same phenotype. Similarly, this set of alleles again located at the same locus are controlling this phenotype, that is this trait that is linked. So genotype in this case can be defined as the set of alleles present at this single locus related with the single trait. Now here we have two alleles controlling the two traits of the same characteristic. Two traits are, one is this round and another is this wrinkled. That is, we are having two traits, And these two traits are defined by these three set of alleles. Either it may be the round or it may be the wrinkled. Round is possible in these two combinations. These two set of alleles are displaying the round trait, and this set of allele is displaying the wrinkled trait. Now here, these two alleles are of two types, depending upon whether they can express itself or not. For example, this genotype, as I have already told you that this set of alleles related with a single trait is called as the genotype. Now this genotype is controlling or displaying the round trait. Okay, and we are having two alleles. This is one allele and this is second allele. These two alleles, this allele which can express its effect, as you know, is called as the dominant, and this allele which fails to express its effect is called as the recessive. Okay, that is, we are having two alleles at this locus, and one among these two alleles is suppressing the expression of this one and is expressing itself and is called as the dominant allele. Another allele that fails to express if it is effective, and that allele is called as the recessive allele. That is, at single locus we are having two alleles. One among them is the dominant allele and another is the recessive allele, and both these alleles are controlling the same characteristic, and these two alleles are related with the same phenotype. Another important point, as I said earlier, that this is the homologous pair, and in this homologous pair, this chromosome has been contributed by the father and this has been contributed by the mother. Now the case may be, if the two alleles coming from the two parents are same, that is, if we are having at this location this capital R, and again at this location we are having this capital R, that is, this individual in which this homologous chromosome pair is present, it has received the same type of alleles from its parent. This condition is called as the homozygous condition, and here you can represent it as capital R, capital R, or it may be the case like this, small r or small r. Then again we will be having a situation like this. These are same alleles, and this condition is called as the homozygous condition. Another case is that if with respect to this locus, this individual has received different alleles from its parents, then the case may be like this, capital R and small r. Then this individual is called as heterozygous. But remember, this homozygous and heterozygous terms are used with respect to this single locus. This heterozygous condition refers to the different kinds of alleles present in an individual at the same locus, not at different locus. That is, for example, if here it is the color of the seed and we represent it by capital Y and small y. Now this allele is different from this allele. Here you cannot say that this individual is heterozygous because it is having capital R and capital Y. Why? Because these are located at different loci. Heterozygous is used for those alleles which are different but are present at the same locus. You cannot apply this heterozygous term for the two alleles which are non-allelic, which are present on different loci. So with respect to the set of alleles, we have either homozygous condition or heterozygous condition. This homozygous condition and heterozygous condition refers to the single loci. And another important thing is that with respect to the set of alleles at the single locus, we are having one as a dominant and one as the recessive. So this is What it means by the set of alleles in an organism on a single locus related to single trait. That is, at a single locus we are having two alleles, and among these two alleles one can be dominant, one can be recessive, and these two alleles can either be in the homozygous condition or it can be in the heterozygous condition. And when it is present in the heterozygous condition, then one can be either dominant or one can be either recessive. When it is homozygous condition, then it can be either dominant, homozygous dominant, or it can be the homozygous recessive. So the set of alleles present at the single locus controlling the same single trait, controlling the single trait is called as the genotype. Genotype has been most frequently used in this sense in the genetics. You can use it in another sense, that the set of alleles in an organism or not on single locus. Now here you can use it in terms of the multiple loci related to two or more traits. That is, when you refer this genotype to two or more traits, then you are actually referring to the multiple loci. That is, what is the set of alleles at multiple loci which are related with two or more traits. Now let me explain it. For example, here at this loci we have the genes which are controlling the shape of seed, and here we have set of alleles which are controlling the color of seed. Now these are two different characteristics. If for example we are having here this set of alleles and this individual is having now this set of alleles, and its shape will be the round, as capital R is dominant. This individual having this set of alleles will be having yellow colored seeds. Now these two are different characteristics. This is the genotype with respect to this, that is single locus and single trait, and this is the set of alleles with respect to this locus related to this single trait. Now you can say that this individual is round yellow, that is its seeds have shape round and color is yellow. Now the set of alleles for these two characteristics will be like this: capital R, small r, capital Y, small y. Now these two set of alleles are referring to the two different loci. This one is referring to this loci and this set of alleles is referring to this loci. So now we have the condition that we are describing the set of alleles present in an individual on multiple loci, on this loci, and this is a multiple loci related to two or more traits. Two or more traits, one is this trait and this is second trait. This is again the genotype, that is it can be this capital R, this is genotype, and here in this genotype we have two different traits. And these two traits are being controlled by the set of alleles which are present on different loci. Okay, now you have understand what it means by the set of alleles present in an organism on a single locus related to single trait. Single locus, it may be like this. This is single locus and this is single trait. Now another case may be the set of alleles present in an organism on multiple loci related to two or more traits. Now in this case it may be like this. This is round and yellow. This is also genotype and this is also genotype, but This genotype refers to the single locus and related to the single trait, and this genotype refers to the multiple locus and it is related with the two traits. Okay, now let us suppose that we have two people on this. One plant is having the round seeds and another pea plant is having the ringlet seeds, and this is homozygous for this round trait and this is homozygous for the ringlet trait. When we cross these two parents, we get the first generation that is having the round trait and the genotype is capital R and small r. This is parent generation and this is the younger generation, or you can say the first filial generation. Now very important point. Here you have two alleles. This is one allele and this is second allele. And here again you have two alleles. This is one and this is second. And here also you have two alleles. This is one and this is second one. These two alleles are present together in this individual. And same is the case with this individual and this individual. In diploid individual, we are having two alleles related to the single trait, and these two alleles are present at the same locus of the homologous chromosomes. This is allele number 1 and this is allele number 2, as I mentioned, and these two alleles together will form this genotype. This is the genotype with respect to this round trait present at the single locus. Similarly, this is a genotype at a single locus with respect to this trait that is related to this trait. And here again we have this genotype present at the same locus related to this trait. Now this is the parent generation and these are its offspring. So this is the next generation. In between these two generations, we have only gametes. And in the gamete, we are not having a genotype. We are having only the alleles. It is the allele that gets transmitted to the next generation. Genotypes are not getting transmitted to the next generation by itself, or it is not the phenotype which can get transmitted from the parent generation to the next generation. It is only the allele that is having continuity. That is, only allele can flow from generation to generation. Genotypes are temporary assemblages of this allele. Now we are having this individual having these two alleles, and this is called as the genotype, and this genotype is formed when it receives one allele from the mother and one allele from the father. Now this is genotype. If this individual reproduces before it dies, then it will transfer these alleles, that is capital R or small r, to its next generation. If this individual is having lifespan of 40 years and it reproduces before its death, then it will transfer these alleles, not genotypes. It will form the gametes, and in those gametes we will be having either the capital R or small r, because at the time of gamete formation these alleles get separate from each other, and these alleles will get now transmitted to the next generation, and in the next generation they will be present as a genotype. As long as this individual lives, these alleles will be present as this genotype. It remains in a specific combination till the organism is living. When the organism reproduces, and at the time of reproduction, this individual transfers this genetic material in the form of alleles to the next generation. So you can say that alleles are having the continuity from generation to generation. It is only the allele that gets transmitted from the parent generation to the next generation. Genotype or phenotype by itself are not getting transmitted to the next generation. The genotype term has also been used in broader sense, and in that sense it may be defined as set of all genes or alleles. That is, what is the set of alleles? What is the complete genetic composition of an individual? At this broader sense, it looks more close to another term that is genome. There is little difference, as genome represents the average genetic composition of the population and genotype represents the genetic composition of the single individual. And second thing is that in genome we have both coding and non-coding sequences, and in genotype we usually refer to all genes which are getting translated into, which are having some specific traits or characteristics. This is the definition of the genotype. Now the second question is how this genotype is related or linked with the phenotype, how closely they are linked with each other. For example, if in an individual with respect to a specific trait we have some kind of genotype, then question is this genotype getting 100% translated or expressed into that specific trait, or there are any environmental factors which are affecting this expression? So let me try to explain it. For example, we have an individual having this genotype. Now this is the genotype of this individual. What it means by having this genotype and how this genotype will get translated into the phenotype. This genotype puts the extreme limit on this phenotype. If this individual is having genotype this capital T and capital D, let us assume that this individual will now be having the maximum height, that will be the 6 feet. Now this individual is tall and is having the height 6 feet. Now this 6 feet is phenotype. This phenotype, 6 feet phenotype, is determined by the kind of genotype. That is, if it is having this genotype, having that as per this genotype, this individual will be having the maximum height, that will be the 6 feet. But this individual will reach to this 6 feet only when it will be having these environmental factors favorable. That is, when environment is favorable, only then this genotype will get fully translated into this phenotype. If the environmental factors are not favorable, then this individual will never reach to this height. For example, if we are having sunlight or temperature, if these two factors are lacking in the environment, then it will be very difficult for this plant to get to this 6 feet height. So if the environmental factors are present, if the environmental factors are available, only then this genotype can be fully translated into the phenotype. This genotype defines the maximum limit an individual can reach. To reach this maximum limit, this individual requires the favorable environmental factors. When there are favorable environmental factors, only then this genotype can be fully translated into the phenotype or specific trait. So what are these environmental factors? Now let us try to understand how many environmental factors are there which can influence the expression of this genotype, and how this genotype, through what stages this genotype can be expressed, get translated into the phenotype. Now, how can we explain the relation between genotype and phenotype? As I mentioned earlier in the beginning of this lecture, that only property we as an individual receive from our parents is the genetic material, and we receive this genetic material in the form of the chromosomes from our two parents. And it is this genetic material present in this zygote that defines all phenotypic features of this individual. All phenotypic features are being controlled or being defined by the genetic material we have received from our parents. And this information about our all phenotypic features is encoded within genetic material. This biological information encoded within the sequences of the nucleotides is getting expressed through multi-step process. That is process involving many steps, and through those steps the information encoded within this genetic material about the phenotypic features is finally getting expressed. As you know that in the zygote we have genetic material within this nucleus, and genetic material within this nucleus is DNA. Now the instructions about the phenotypic features are stored in this DNA. Now what this DNA stored in this nucleus will now form an intermediate molecule. That intermediate molecule will be transferred to the cytoplasm, and then in the cytoplasm it will form another molecule, and that molecule will perform the functions. But here it is important to mention that the first environmental factors which is surrounding this genetic material is the cytoplasm of the zygote itself. That is, it is the first external environment of this genetic information. In addition of the cytoplasm surrounding this genetic material in the nucleus, other cells also influence the expression of this genetic material. Then we have external environmental factors which will influence this expression of the biological information. And finally, by the coordination of all molecules, by the coordination of all internal factors, by the coordination of all external environmental factors, finally this biological information gets expressed in terms of the phenotypic features of an individual. But the base of these phenotypic features is this genetic material which each individual is receiving from their parents. As I mentioned that the genetic material present in the nucleus is in the form of the DNA. Now this DNA will first form intermediate molecule that is called as the mRNA. And this mRNA will get transferred from the nucleus into the cytoplasm. And this process, as you know, is called as the transcription. It is the first stage of the expression of the biological information encoded within this DNA molecule. That is the formation of this intermediate molecule, that is the mRNA. And this mRNA will now lead to the formation of the protein. The formation of protein from this mRNA is called as you know, translation. This is the second stage of the expression of the biological information. First is the formation of the mRNA from the DNA, that is the transcription. Then we are having the formation of the protein from this mRNA, that is called as the translation. Once this protein is formed, now the actual function will be performed by this protein. Execution of the function is as per the instructions encoded within the sequence of the nucleotide is performed by this protein. This protein will perform the functions in the cytoplasm, or in many cases this protein will get released into the surroundings and will influence the activity of the neighboring cell, because neighboring cell might be having some kind of receptors, and these receptors will receive the signal from these molecules, and accordingly now this cell will modify the structure and function as per the instructions or as per the information received through this molecule. Now first we have this genetic material within the nucleus, then we have this cytoplasm as the immediate environment of this biological information. This biological information encoded within the DNA is first getting transcribed into the mRNA. This is the first level of the expression. Then when mRNA is formed, it forms the protein, and this protein is formed from the mRNA through the process that is called as the translation. These two steps are the initial steps of the expression, gene expression. Here it is important to mention that replication, another function associated with the DNA, is not considered as a part of the gene expression, because replication is required when cell is about to divide, that is when the division is to be occurred, then this DNA forms the exact copy of itself so that it can be equally distributed into the two cells. So replication usually is not the part of the gene expression. Once the DNA forms the mRNA, then it is certain that now this gene is going to express its effect. And this formation of mRNA through the transcription is the first stage. Then formation of the protein through the process of translation is the second stage. So we have transcription and translation, first two stages for the expression of the biological information or the expression of this genotype as a set of alleles present in this biological information. When one is these proteins are formed, one is these tissues are formed, one is this organs or organ systems are formed, and one is this individual is formed. Now this individual will be exposed to the environment, or you can say the external environment. Now everything around this individual is external environment. It can be the physical factors like the climate, like the geographical position where it lives, or the psychological factors, the family background, the society in which he lives. These are the environmental factors, and these factors will definitely determine how this individual is going to develop. What will be its future phenotypic features, what will be its structural features, how this individual will function, and what will be its behavioral aspect. So all these factors will be determined by these external environmental factors and internal environmental factors at this molecular level. So just having the genotype is not sufficient to have the phenotypic features. We require thousands of external environmental factors, we require thousands of molecules, and we require thousands of other factors in the neighboring cells, and it is the result of this complex molecular dialogue at the molecular level, then complex interaction between the cells and complex interaction between the tissues and the organs and the organ systems, that ultimately leads to the expression of this genotype into the specific phenotype. So here we can conclude that genotype refers to the set of alleles with respect to a single trait located at the single locus, or it can refer to two or more traits located at multiple loci, or it can represent a set of all alleles present in an individual. And it is this genotype which gets finally expressed as the phenotypes. Genotype is not sufficient to have the phenotypic features. There are thousands of environmental factors, both internal as well as external, which influence the expression of the biological information encoded in these alleles.